Write diffusion as movement of molecules, and in bracket it's better to mention which molecules are moving by the process of diffusion. So you will get 1 mark in Cambridge if you are going to write this line. For this one statement sentence, there is 1 mark. Osmosis, it's the movement of water molecules. So water is must here. For this point, you will be given 1 mark. And in active transport, you also have to write down that it's the movement of molecules. And in bracket, mention the molecules which move by the process of active transport. So 1 mark is for the molecules which are moving. Second mark in Cambridge in mark scheme, that is for from higher concentration movement of molecules from higher to lower concentration. You will be given second mark here. Same is done in water. 1 mark is given for higher concentration to lower concentration, and here it's given 1 mark. So 2 marks are for the movement of molecules from higher to lower concentration. Third is for membrane, right? It is with or without membrane. You will be given 1 mark here, 3 points. And this is also with partially permeable, 1 mark here. So 3, and it will also be marked here. So 3 marks. Next is using energy. It is very important here. Here without using energy, and it's also without using energy. Sometimes this is marked, but sometimes it is not marked. But if you are going to write this, it means that you would have written 4 valid points according to the mark scheme. So you can get 4 marks in this one definition, right? So this will be giving you another mark, means we have gained 4 marks. So its definition by writing all these points which I have underlined or marked. For example, you are given another mark. How if you're going to write more than one example, still you will be given just 1 mark. Sometimes writing more examples can give you more marks. But if you're going to write 2, 3, 4, whatever the number of examples you're going to write, you will always be given 1 mark. So it's better to write down one example, right? For 1, you will be given 1. 1 here, 1 here, and 1 here. So now you can see this definition and only this example is going to give you 5 marks. Here it's also going to give you 5 marks, and here it's also going to give you 5 marks. So if I ask you to write down the processes through which the molecules move across the cell membrane, and you are going to write about diffusion, osmosis, and active transport, and you are going to write down all these points which I have marked here. So it means that you have attempted the question of how many marks? 5, 5, and 5. It means that you have attempted the question of 15 marks. Some points might not be in the mark scheme, so it means you can easily get 10 marks if you're going to write only these points which I have mentioned here. Okay. Next is the type of solution. Solution always has a solute and a solvent, and there are 3 types of solution. One is hypertonic, isotonic, and hypotonic, right? This is hyper, this is iso, and this is hypotonic. Tonicity is used to describe the concentration of a solution, right? Hypertonic, it has high concentration of solute, right? Solute is in high concentration and solvent is in lower concentration. It has high solute and low solvent. It is also known as the concentrated solution. This is concentrated solution because it has high solute. Another is the isotonic. Isotonic has same concentration of solute and solvent. Hypotonic has a low concentration of solute. It is opposite to hypertonic, and high concentration of solvent. It's also known as the dilute solution. These are the 3 types of solution. Now we are actually comparing these solutions to some other solutions. We are saying that this is the hypertonic. It is hypertonic as compared to hypotonic. Means we are comparing this solution with something else. But if we are talking about the condition of the cell, let me draw here. If I say that this This is the cell, this is the beaker, and beaker has hypertonic solution. We are actually comparing it with something. That's why we are saying that it has high solute and less solvent. For example, this is any cell, animal cell, which has been placed into this. This is the animal cell. Animal cell will be hypotonic. This cell will be hypotonic. This is the hypotonic. If the solution outside is hypertonic, so this will be hypotonic solution. Inside will be hypo. Now we are comparing this outside solution with the inside. That's why we are saying that this is the hypertonic and inside is the hypotonic. And opposite will happen. If outside there will be the hypertonic, inside there will be the hypertonic solution. Now to remember, hyper has higher concentration of solute and lower concentration of solvent. Isotonic has both solvent and solute are in same concentration, and hypertonic has lower concentration of solute and higher concentration of solvent. Now what happens if the animal cell and plant cells are placed into these solutions? This is the hypertonic, isotonic, and hypertonic. If the plant's animal cell, first let's discuss the animal cell. If animal cell is placed into the hypertonic solution, hypertonic means that outside there is more water. Okay, hypo means that, come again please.
This is red blood cell.
These are red blood cell, or you can place any animal cell. Same will happen to all the animal cells. If you're going to place them in hypotonic, hypo means that there is more water outside, right? Here you can see there is more water outside and there is less water inside the cell. This is water and it has more water outside. So water will move from high concentration to lower concentration by the process of osmosis. Right, so water will move from outside to inside cell because inside there is low concentration and outside there is more concentration of water. So it will move inside by the process of osmosis, and ultimately what will happen? The cell will swell first and ultimately it will burst, which will lead to the death of the red blood cell. Isotonic, no change occurs. It's almost equal because inside the concentration is also same and outside the concentration is also same. So the same number of molecules are moving inside and same number of molecules are moving outside. So it has no effect. Its cell will remain be normal. Hypertonic reverse will occur as the hypertonic solution has low concentration of water and inside there will be high concentration of water. Upward arrow is for high concentration and outside there is low concentration. So water will move out again by the process of osmosis. So what will happen in this case? As water is leaving the cell, so cell will shrink, right? It will shrink and ultimately it will also die. So in this case, we have seen if the animal cell, any animal cell, white blood cell, red blood cell, liver cell, kidney cell, they are placed into hypotonic and hypertonic solution, death occurs. In hypotonic, water moves inside, cell swells and it bursts, and in hypertonic, opposite happens, the cell moves outside and water moves outside and the cell shrinks and it also dies. Now we are going to see what happens to plant cells if they are placed in these solutions. If we are going to place plant cell into hypotonic solution, so what happens in isotonic? No change occurs, cell remains flaccid. Flaccid means normal, right? We are going to discuss hypotonic and hypertonic. If the plant cell is placed into hypotonic solution, so what will happen? In hypertonic solution, water will move inside. Because of high concentration of water outside, water moves inside by the process of osmosis, right? Now I'm just going to draw this plant cell here. This is the plant cell, cell wall, cell membrane, and vacuole. Water moves inside by the process of osmosis, right? And this cell also swells, but it do not burst because of cell wall. This plant cell do not burst because of the cell wall, and when water enters continuously enter into this cell swells, and that is known as the, we say that cell is turgid, because sometimes the plants look fresh and this freshness is because of the movement of the water inside the cytoplasm. They do not burst because of the cell wall. Please stop writing on the screen. And
Sorry,
cell,
it was my mistake.
okay, the cell is said to be turgid. Cell will be turgid. Okay, and the process is known as, this is turgidity, right? As the water is moving inside, so this water will exert pressure on the walls of the cell, and this pressure is known as the Turgor pressure. Here there are three terms which you have to learn. One, water will move inside by the process of osmosis. We know, as we have seen in animal cells, they swell and they burst, but plant cells do not burst because of the presence of the cell wall. Water moves inside and water exerts pressure on the walls of the cell. That pressure is known as the Turgor pressure, and this process is known as the turgidity. And the cell which appears to be fresh, swollen up, that is, the cell is known as the turgid. The water helps in turgidity of the cell, and this turgidity is also because of the cell wall. Next is, if the plant is placed into the hypertonic solution, what happens? Let me draw here. Hypertonic solution, this is the in hypertonic, water will move outside. This is the plant cell, right? And what happens in plant cell? Water move outside because of high concentration by the process of osmosis, and it will continuously occur. What happens? The cytoplasm of the cell is shrinked. Not only whole cell shrinks, I have drawn here two. There will be only one line. Only the cytoplasm of the cell shrinks, and this is known as the plasmolysis. Plasmolysis is the shrinking of the cytoplasm. Plasmolysis, it is the shrinking of the cytoplasm. Only cytoplasm is shrinked. This is shrinking of cytoplasm, right? But this cell also do not die because of the presence of the cell wall. If continuously the cell is placed into the hypertonic solution, hypertonic solution, what will happen? Water will continuously be moving outside, and ultimately it will also die. Shrinking of the cytoplasm is known as the plasmolysis, and the cell which has undergone plasmolysis, that is known as the plasmolyzed cell. Okay. Now, if we are going to place it in water or hypotonic solution, which has high concentration of hypotonic solution, which has high concentration of water, so water will move inside, and it will move in cell, and cell will be normalized again. And that cell is known as the deplasmolized cell. Only plasma cell, if the plasmolized cell is placed into the water or in hypertonic solution which has high concentration, the water will start moving from outside to inside, and that will help cell to regain its original position, original shape. That cell will be known as the deplasmolized cell. Deplasmolized means that its plasmolysis has been removed. If we are going to place a normal cell into this hypotonic solution, that will be turgid. First, this plasmolyzed cell will be deplasmolized, and then it will also be turgid if more water will continuously move inside the cell. Right? This was about the solutions and osmosis, and this chapter is also completed. Any problem? Guys, if there is any problem?
No, miss.
Okay, this was a very small chapter. There were only 3 to 4 terms. So I will send you these notes as it is. I have written here into the group, and then you can note them down on your notebooks. Okay, time only 1 minute is left. I'm going to write here deplasmolized so that it should be easy for you to note it down when you will be copying it on your notebooks. So this cell will be deplasmolized, and the process will be, as it's the plasmolysis, so the process will also be deplasmolysis. Hope so you will be able to write it down.